Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
So the title is sort of ambiguous. It could be either programming languages for life, like living systems, or it could be programming languages for life, like life sentence, with programming languages. And actually, both meanings are intended. So I'm going to start with the very early days of Luca and kind of try to cover the history of computers and programming languages from very early on. When Luca was 2 years old, that computer was built at the Weizmann Institute of Science in 1956. Actually, it's just a part of it. It's another part. That's another part. Here are a few more parts. And that's the Ziskin building, which was actually the Weizmann Institute of Science at the time, which had a huge basement, and the entire basement was filled with this computer. Since then, we've seen two important trends. First of all, since Luca was 2 years old, he started slowly growing up and gaining weight. And since that time, computers started decreasing in size and weight. And I tried to calculate when the two curves crossed. When was Luca's weight higher than his computers? And you know what is the answer? I think I figured it was around when he was 25 or 27. When he used the lift machine, he was still lighter than his computer. But when he switched to the Mac, I think Luca's weight was higher than, right? So you're about 25, 26, something like that. So for the first 25 years of Luca's life, the computers were heavier than him, at least those that he was aspiring to use. Okay, that's one bit of important history to know. When Luca was, I think, 3 or 4, let's see, yeah, FORTRAN was invented by John Bacchus and his team. When I asked Luca about FORTRAN, his facial expression was not extremely positive. And I must admit that when I studied FORTRAN, this was my first programming language. Luca was luckier. His first programming language was Simula 67, as he told me. So he had a much better early life than mine. I had to struggle with FORTRAN, and I couldn't believe why people would invent such an ugly, at least to my aesthetics, construct. But things got a bit worse later, you will see. Anyway, for those who are not familiar with how you program in FORTRAN, then you would use these devices, and the way you would debug, you would feed the card on one end of the card reader and copy part of it, and then type the part you want to correct, and then press the copy button for the rest, and that's how you would fix it, if you had an access to such a machine. If you didn't, then what you would do, you would fill these coding sheets by hand, and give it to someone else who might have time to type them for you or not. And then you would program. That's how you would program. And when I was learning, it was in high school. So the debugging cycle time was 1 week. The teacher would take the coding sheets, go to the nearby university. They would type the cards, run it. He will bring it next week. And that was our debugging cycle for FORTRAN programs at the time. So the next important milestone was a couple of years later, the first trend of programming language design by committee, which I think became extinct after a while. Anyway, Luca also was exposed to this language, and I also worked a little bit to earn a living programming in COBOL. I was an employee of a subcontractor of Bank Hapoalim, which is a big bank in Israel. And do you know what the programmers in the Bank Hapoalim had on their desks? Any guess? Nothing. They had nothing on their desks. But you know what they had behind their desks? They had shelves with boxes, like big boxes, with cards in them. So every programmer had these 2,000, 3,000 lines of code, which he was responsible for, and it was organized in boxes, big boxes, which they would carry to run or debug or compile to the computer room. And on each box, there was, with a marker, a very large diagonal line. Anyone knows why the hell would they put a diagonal line on the box? Luca knows. If two programmers bump into each other in the corridor, each carrying his box with the cards, what happens? And the cards fall on the floor. How would they sort them back? They just put them, and according to the diagonal line, they would put the cards back in order. So that's the legacy of COBOL programming and these lines. Okay. Moving on, I think a revelation for everyone was Pascal, including myself, in the 1970s, where suddenly you'd see that the programming language does not need to be ugly, and for me it was even a profound revelation. The Turing Award lecture by John Bacchus in 1978, but Luca says that he has been already converted to ML before that, so for him this was a minor event. Anyway, for me this was seeing that programming does not have to be separated from clean intellectual mathematical thinking, but can actually embody it. And the examples in the paper I thought were extremely fascinating. My intellectual environment, my teacher was at the time was Amir Pnueli, who was a very strong proponent of imperative programming languages, and he looked at this and said, well, this is a nice toy, but it's not for real programmers in real life. And I think these two camps still, the legacy of these two camps is still living today, and probably resolved in the future at some point, or not. I will not get into this thing, but Bacchus repented for his FORTRAN sins by offering this Turing Award lecture, but still 35 years later, FORTRAN still won a Nobel Prize, all these people are really avid FORTRAN programmers who still use FORTRAN. Two of them are actually Weizmann Institute alumni. They did the beginning of the Nobel Prize work they did at Weizmann with their advisor who died, so he was not eligible at the time for the prize. But anyway, so FORTRAN still lives on despite of all our reservations from it, and even got a prize. So what happened next? I think my intellectual, well, I should say that both biologically and academically, Luca and I are more or less of the same age. But our paths we've gone on parallel paths, actually surprisingly parallel. I'll show you a couple of anecdotes in a second. For at least 20 years, kind of minding our own business without interacting with the other, but really in parallel. And for the last 15 years or a bit less, we've been, our paths have crossed. So it doesn't look exactly like a chromosome, but still that's kind of a depiction of history. So I'll do a fast forward for the first 20 years just to set the stage for the main part of the talk, which will discuss the work that we have been doing in collaboration. So this is black because these are the said 20 years without Luca, said 20 years of my life without Luca. So I'll just say a couple of things. First of all, it turns out that both of us started our PhD work in AI and in the AI department or section, and then moved after some initial, for different reasons, we moved to computer science part. And we both finished at the same month, on April '82. We both got our PhD in different continents, but the same too. And we both mostly enjoy Bach music. So I think these are three important facts. Even though we're living separate lives, you see that there are some similarities and relations. So while Luca was doing his programming language work, I started in the area of inductive inference, trying to work on inductive inference of theories from facts. And, but the first kind of point of almost intersection was when I heard of Prolog, which was developed by David Warren, Fernando Pereira in Luca's alma mater, I think while he was there in Edinburgh. And I realized that, I should say that the natural implementation language for what I was doing at Yale at the time, both technology-wise and environment-wise, was Lisp. But I couldn't, for some reason, connect to this language. And when I heard of Prolog, I said, that's what I need. So I wrote to David Warren, and he replied to me that the only copy of Prolog in the United States, the Prolog compiler for the DAC-20, is in New Mexico Tech. And he gave me the name, address of the person. So I wrote to him, and he sent us these big tapes with Prolog, and we installed it. And apparently, he forgot to send us the user manual. So whatever I type, the Prolog interpreter would say no. No. He typed something else, said no. So I said, what's going on? So I said, I know what I'll do. I typed true. And he said, yes. So I said, actually, there is some intelligence in the machine. Now we have to figure this out. So after a while, we also got the manual, and I did write this inductive inference system in Prolog, and got sucked into programming language research, and wrote a textbook about it, and worked on concurrent programming, concurrent logic programming, collaborated with the Japanese people. These are all happening while Luca was doing other things, great things that we've heard about in the last couple of days. Started an internet software company that was doing real-time distributed instant messaging and communication on the very early days of the internet, before it was popular. Sold it to America Online, then bought it and sold it to IBM. And then while doing this, I sort of was trying to figure out what I want to do with my life, and I realized that this is not what I want to do in my life. So I went to the Borders bookstore and bought this thick and heavy book called Molecular Biology of the Gene, and started reading it. And decided that what I want to do for the rest of my life, at least for the next phase of my life, is not to continue with this internet crazy, world. They would manage well without me, but to go back to Weizmann and start something new, research on something which was not clear, molecular computing. I thought building computers from molecules. So my main interest was molecular computing, which is trying to build computers from molecules. When a computer scientist sees a new media, the knee-jerk reaction is, how can I make this compute? So that was my knee-jerk reaction with molecular biology. How can I make it compute? And that was my main research effort and thrust at the time. But I had this sidetrack into molecular programming. I had this PhD student, Aviv Regev, who was formerly a PhD student in Tel Aviv, but she came to me and said, look, I have this idea that I want to use computer programming languages to describe molecular systems. So I said, well, I want to use molecular systems to build computers, but let's talk and see what happens. And we talked and talked and talked, and we converged on the idea of using pi calculus for describing this, and the question was how could we make it work? And I said, okay, let's look for pi calculus implementation and use it. Well, it turns out we couldn't find any such. And then I remember that I wrote, actually, I forgot to show it, but several years before, I wrote a paper, before my internet detour, I wrote a paper on embeddings among concurrent programming languages, which was my kind of excursion into category theory. And I showed, among other things, that pi calculus can be embedded in concurrent Prolog. And we did have a concurrent Prolog system, collecting dust at Weizmann. So I said, okay, pi calculus can be compiled to concurrent Prolog. Let's write an implementation of pi calculus in concurrent Prolog and use it. And And we did, and lo and behold, it worked. So this led to this initial work of Aviv. And basically, the key insight was that there is a very natural analogy between process algebra, computational entities, and molecular entities. That molecular species are analogous to process species. Molecular population is a system of concurrent processes. Complementary motifs and types are complementary input and output occurrences on the same channel. And motif occurrence in a molecular domain is communication offers on channels in processes. So, and then all the operational semantics followed. So really had this very naive insight that there is a very strong correspondence, we should make it work. And we started making it work by writing examples of code in PyCalculus and debugging it and seeing that it actually behaves like the molecules that we saw. And like the molecules, we knew the molecules behaving. And we wrote a couple of papers with Bill Silverman, who was the implementer of the system, showing that you can do various things. But it was very clear that it's really just the first step, that molecular systems are stochastic and PyCalculus is not, and we should have a stochastic version of PyCalculus. So we searched and found that actually there is a stochastic version of PyCalculus by Corrado Priami, who is here and somewhere, and discussed it earlier today. Is he here?
He's gone.
Oh, he's gone? Okay. So I can say what I want about it. Right? So anyway, so we opened a discussion with him and basically implemented stochastic PyCalculus on top of concurrent Prolog system we had as an extension of the PyCalculus implementation using the Gillespie algorithm and tried to run it. And it behaved completely bizarre. And then we realized that the stochastic rules that were in the definition of stochastic PyCalculus as made by Corrado were very good for computer networks, but were completely incorrect and useless for molecular systems because we want the probability of interaction among molecular systems to be a function of the number of molecules in the system, basically. And the original PyCalculus had a different definition. So we revised it so that it really captures the classical kind of probabilities of molecular communication. So the probability increases the more offers there are for molecules to interact in a particular way. And we got the correct behavior and simulations, and that was a really very nice. You can see here the graphs of various things we implemented. We were very happy with it. So then I had the occasion, I forget how and why, but I really wanted to visit Robin Milner and tell him about what we're doing and have his insight and feedback. And so I told him that PyCalculus that is alive and well and kicking, and we actually use it, and showed him all the examples. I think I gave a talk in his group seminar. And in our discussion, I said, look, what's really missing for us now is some sort of structure. The PyCalculus is like a big soup of molecules. And in biology, you have all these compartments, and things are happening locally, and molecules can move from one compartment to another, and all biology is about that. So I said, look, next door or next building, I forget, there is Luca Cardelli, and he's done this ambient calculus. Maybe what he's doing is relevant to achieve that. So same building or?
Next building.
Next building, yeah, I remember. I had to cross. So I crossed the street or whatever to the next building, visited Luca, and again started this talk from the beginning, and told him all about this and about compartments. And he said, yeah, it's interesting. And I think one of my main scientific achievements in my scientific career, which I'm most proud of, is that event, because I basically sucked one of the great minds of computer science to think about biology, and you've seen what happened as a result. So Luca got interested in more and more and more, and basically this effort took off. I'll tell you many of the consequences you've heard of, and I'll just mention some of them. So the first result was this joint publication, my first paper with Luca from 2002, 3, something like that, and Aviv Regev and which became a chapter of Aviv's thesis. And it had many nice examples in it, many figures of different molecular systems, and that can be nicely implemented using bioambience, which included also the pi calculus as a subset for direct molecular interaction, and also the notion of compartments. And of course, we implemented it and simulated, and we were very happy with it. And what happened then? Aviv graduated. This unfortunate event happens for most graduate students, most PhD students, and left. And we had this dilemma, what to do with the project. I told her I had enough, I ran enough large-scale software development projects in my life, and I'm not willing to have another one. So I'm not going to do what it takes to make this system for real, a real usable, programming system with user interface and manuals and all that. She is welcome to do that. And she said, no, I want to have another direction in my career. Well, I'm not sure she said it so explicitly, but in practice, she went to her postdoc in Harvard and basically told me she wrote up the papers, but did not want to undertake this as a large-scale software project for the rest of the career. So this was just kind of left up in the air, and we're extremely fortunate that Andrew Phillips and Luca decided they want to take this forward, and basically re-implemented the stochastic pi calculus on top of using their own engine, and it took a life of its own, and is now a robust and usable software system that many people can use, and that's a great outcome of this work. Basically what happened since, and actually I gave this talk a year ago, almost not knowing that I'm going to have this occasion to give it to Luca. I basically tried to tell the world that there is this great computer scientist who is doing amazing things in biology. They should just look at his website. So I think you can probably tell when how late is this web page, but it's somewhere around a year old or 10 months old. You can see that Luca has really spread his interest on almost many aspects of computational biology. I find it completely amazing and unbelievable that he can do all this amazing research in so many different directions, and the results are here on his website, nicely organized, and you can see that many different aspects of biology and computation are addressed by his research. The list goes on, and since a year or 10 months ago, when this snapshot of his website was taken, many interesting papers, more interesting papers and important papers emerged, including the most recent one, which is really beautiful, on the relationship between structure and function of circuits, which is really, I feel it's a really fundamental piece of science that will have impact for many years to come. So the best for Luca is yet to come. That's what I'm trying to say. I just want to highlight one paper which came out, I think, a year ago now, or last year. And for me, this is, personally, it's an extremely gratifying paper. Because, at the time I was working in two directions on molecular computing, trying to use molecules to build computers, and on molecular programming, trying to use programming languages to describe, simulate, analyze, design molecular systems. And I had two PhD students working on this, Aviv Regev and Yaakov Benenson, and they were friends, etc., but they were completely unrelated at the time research directions. I don't think they even referenced each other's work because they were just, on the face of it, unrelated. And they both graduated and continued, and this was more than almost a decade ago. And since then, these two fields evolved and developed. And this paper is really the first paper that kind of merges them together by bringing Lucas, Andrew Phillips, and their team further developments in molecular programming on systems for describing the behavior for programming languages, for describing and simulating the behavior of molecular systems, and they work on molecular computing by some groups led by George Zillig and others. And basically, the molecular computing people were trying to build computers so complicated they couldn't design and simulate them by hand, so they needed the molecular programming people, Luca and Andrew Phillips, to help them design and simulate the molecular computers they were trying to build. So this paper is really the culmination of an important milestone for these two fields, where molecular programming was used to design and simulate, analyze, debug, predict the behavior of a molecular computing system. And there is a little cherry on top because the algorithm, the key algorithm that they were trying to implement, that they implemented successfully, which is the most complicated distributed algorithm implemented using molecular computing, is the approximate majority algorithm. It has been discussed earlier today in the context of the cell cycle switch. And the designer of this algorithm is Dana Angluin, which was actually my PhD advisor. So it's like all pieces of the puzzle fell in place, and it's really an important and beautiful piece of work. And for me personally, it's like a little celebration. Okay, so that's first chapter of my collaboration with Luca. What time it is? I have another 15 minutes. That's great. So Luca, as you've seen from his website, Luca went on to many different directions in biology and computing. And since Aviv left my group, I didn't continue personally research on molecular programming. And my research interest drifted into other things on the border of computer science and biology. But you will see again that Luca and I had another interaction on that. So bear with me for a few minutes. So another project that started just because we were there and we were on the board of computer science and biology was trying to understand cell lineage trees. So we all know about strings, which bioinformatics people use to describe DNA molecules, and it's an important abstraction. And in data structures, the next structure you learn after you learn strings, you learn trees. So trees are also important. And in particular, trees can be used to describe the cellular history of multicellular organisms. So here we see the cellular history of a worm called C. elegans, which starts like any other multicellular organism from a fertilized egg and goes through consecutive cell divisions, binary cell divisions, and cell death until the organism becomes, until any point in time. And this depicts the growth of C. elegans till maturity. And the rooted, labeled binary tree can capture this level of abstraction of the history of the organism. Now, many other things can happen, of course, and happen during organism development. Cells change, they interact with each other. All these are abstracted away. And the cell lineage tree just captures the history of which cells were and how they divided and their topology and maybe their type, etc. So science knows the cell lineage tree of, I should say, that this is a C. elegans. Now, science knows the cell lineage tree of one organism, this. And for more complex organisms like mice or us, science is still very much in the dark. Now, each of you, one of the reasons is that the organism is much more complicated and large. Each of you, really, your walking self is really the leaves of your cell lineage tree. Basically, your cells are the leaves of the cell lineage tree, which is about 100 trillion nodes big. Okay, so I hope it's not a shocking news to anyone here, but each of you started your life as a single cell, as a fertilized egg, which is the result of the fusion of your mother's egg and your father's sperm. And since then, that cell underwent binary divisions until you became yourself. And God knows a cell lineage tree, which is this mathematical entity which represents your cellular history. And we would like also science to know that, not just God. And the question is how to do that. And my research has been on that since. Okay, that's just the structure of the tree. I don't think you need this information. And that's what I said. And basically, we had an insight that cell lineage trees can be reconstructed from the noise of the machinery of DNA replication. The dogma in biology is that each of us has a different genome, the unique DNA that makes us what we are specifically, but all the cells in our body have the same genome. It's our genome. Our unique genome is in all the cells. So this is true to a first approximation and is false to a second approximation. And the reason it is false is because DNA is replicated extremely accurately when cell division occurs, but not absolutely accurately. So there is some leftover noise in the division process, which every cell carries some noise which is unique to that particular cell division. And if we just collect all the noise, all the errors that occurred from the fertilized egg up through all divisions up to a certain point in time for each cell, then each cell has a unique genomic signature, genomic signature which is unique to the organism with extremely high probability, which is just all the errors accumulated during cell division. And of course, in some cases like cancer or immune system, the mutations are even more profound, but I'm talking about normal human cells. And so that's one thing. So each cell in our body has a unique identifier. It's the accumulation of all mutations from the fertilized egg unique to that cell. And also the distances, the Hamming distance between these strings, if you will, really reflect the distance in the lineage tree. So if two cells are only one division apart, they have a few mutations that separate them. If there are 20 divisions apart, they have many mutations that distinguish between them. And what we showed in our first paper on this subject is that these mutations are sufficient to reconstruct the human cell lineage tree with extremely high precision if we knew them. Okay, so what does it mean if we knew them? Let's say a person donated his or her body to science after death, and we analyze all the cells in the body. So we have to sequence every cell, find the mutations in every cell, and then we could reconstruct the human cell lineage tree completely. So we have about 100 trillion cells. Sequencing the whole genome is about $1,000. So for one person, it's $100,000 trillion, and we could have the answer. So that's a bit extreme, I think, even for Microsoft's budget. So we need to do other things. And basically, I'll tell you a little bit what we've done. But first, maybe before spending $100,000 trillion or even a fraction of it, we should see why we want to do this. And the answer is that many fundamental questions in biology and medicine are about the structure, dynamics, and variance of the human cell lineage tree. And I can say this I've been in this business, in this kind of, around this research question for more than a decade, and I can say it with ever-growing certainty that if you go from one field to another of human biology and medicine, people argue about, fight about, don't know, ask the questions. They don't use this language because it's a computer scientist language, but what they really ask is about the structure, dynamics, and variance of the human cell lineage tree. And just a very simple example, in leukemia, we know that when we treat people with chemotherapy, some of them recover and some of them look as if they are cured, and then after a year or 2 years, suddenly they have leukemia again. So the question is, where does this relapsing cancer come from? And basically, there are two possible answers. One is it's like this, that the chemotherapy killed almost all the cells of the cancer, but not all of them, so some escaped. And then the new cancer is just a descendant of this. We see it on the left, on the left side of the figure. Or another possibility is like this, that maybe chemotherapy killed all the active cancer cells, so all those that are dividing quickly, and the distance from the root really represents the number of divisions. But there are some other cells that were not killed by chemotherapy, and maybe because they're dividing slowly, because chemotherapy really targets quickly dividing cells, and the new cancer emerged from these cells. And the question is, it's a very simple question. What is the case? And of course, if we can know the cancer cell lineage tree, we can answer this, and it has major implications for therapy. Because if it's this case, it means just give more chemotherapy. Maybe you'll kill also these remaining cells. Just give chemotherapy should be 10 times strong or 100 times strong to kill each and every last cell. But if it's this case, it means, look, chemotherapy killed all the quickly dividing cells. This cancer started from slowly dividing cells, so no more chemotherapy will never kill it. You should find something else. You should identify these other cells and kill them as well. And our experiments really supported the second case in collaboration with Rambam, that indeed the cells in the relapsing cancer were more shallow than the cells in the primary tumor, and therefore could not be their descendants, logically speaking. And we had other evidence suggesting that they are closely related to what's called cancer stem cells, which basically has the property that even though they divide very slowly or divide rarely, they have the potential to create cancer, if they choose to. And this is indeed what happens, and therefore therapy should try to target also cancer stem cells. Okay, so this is just a small example why this is important. And And we've built a system to try and analyze single cells and mutations and reconstruct. I'll skip all that. And we're still building on that. I'll skip on that. And we're still trying to build this pipeline using the latest technology of microfluidics and next-generation sequencing. That's really the major project in my lab, and I'll skip that as well. And I'll come to my second paper with Luca, which basically arose out of the need to calibrate and analyze and improve the algorithms we use for cell lineage reconstruction. Because what are these algorithms? We take mutations from the leaves of the tree and try to reconstruct the lineage tree using standard phylogenetic algorithms to do that. But how do we know if we're doing well or not? Biology doesn't know the tree, so any tree we say, they can believe it or not believe it, or how can we prove that what we're doing is correct? And there are basically two avenues to do that. One is to go to biological trees that you can reconstruct, that you know exactly the structure, either because it's a system in biology, in mouse biology or human biology, that people know exactly their structure. For example, in colon crypts, it's the case, and we were able to show that we reconstruct the lineage trees of colon crypts correctly. Or you kind of manually grow cells and allow them to divide and separate them and have a collection of cells that you know exactly which cell begot which cell, and then take the DNA from them and see whether you can reconstruct the tree that you've grown. And this is presently very extremely labor intensive, and in our first paper from 2005 we did that and showed that we can indeed reconstruct cell lineage trees, coarse cell lineage trees, but it was really one example. So what you really want, you want a language that can generate many types of trees in a programmatic way with mutations, and then computationally, and then take the data from the leaves of the tree, reconstruct the tree, and then compare the tree that was generated by your software to the tree that you've reconstructed. So, as I said, people who are infected with the programming language bug, they see always an opportunity for a new programming language everywhere they look. So for us, it was as well. We saw an opportunity for a new programming language to describe these processes. So, first of all, in these populations, you can see you can go for a deterministic solution, which is solving differential equations, or for a stochastic solution, which simulates the species of the cells that you have, or the entities that you want. And for us, it was clear that we must go the stochastic route because we want to simulate precisely what's happening, not solve the differential equations. And this is an example of the top one is a predator-prey system. The bottom one is just a system that grows up to a limit and then stays there stochastically or deterministically. And the first thing we'll do is let's look at what exists. And two natural candidates were chemical reaction networks and stochastic pi calculus. Now, chemical reaction networks are standard tool. I have to finish soon. Chemical reaction networks are a standard tool for describing these, but they're not good. I'll skip the long justification why they're not good. I'll just say that they're not good because you cannot track histories and lineages. You just have these anonymous populations of entities that interact. And process algebras are not good because they are too complicated and unnecessarily detailed. So we really wanted a language which is much simpler and easy to describe these lineages. And we looked at stochastic tree grammars, which are basically context-free grammars with probabilities, which is a natural choice. And then the tree structure will just give us the trees. And we played with these for a while. And we were able to get, reasonably nice behaviors of stem cell differentiation, etc. As you can see from here, these are reminiscent of the things you get from stochastic pi calculus, but you also get the tree structure, which is very important because you want to understand the lineages. But the bad thing about it is that the behavior of the entities, because it's context-free, is very unstable. So you can get, in different stochastic simulations, radically different behaviors of the population, which if you average all of them, you will get the correct behavior, but each individual behavior is highly noisy and unstable. So our solution was to make a little twist to stochastic tree grammars and not change their context-free nature, which makes them very simple and elegant to design, but add a little environment component, which basically you allow the probabilities to be not the tree structure to be controlled, but the probabilities and rates to be controlled by environmental parameters, typically the size of the various populations. So if you do that, you can see that you can, with a very easy twist, like here, by adding the number of A elements into the probability, you can get a very nice stochastic control. So you still have a simple context-free grammar for either A can either divide or die, but the probability is a function of the number of A's, and if it grows above 100, then the probability of dying increases, and if it goes below 100, the probability of splitting increases, and therefore it fluctuates very nicely between these two. And basically we took off with this idea, and because I'm out of time, I'll just skip through this, showing you that we wrote in this paper. These are all figures from this nice paper we published with Luca. We showed that you could write very elegant, simple programs for the behaviors that you want, which is really what programming languages are for. And we have this simulator to execute these programs, and also tools for for their analysis. And you get both the dynamic behavior in terms of process counts and also the tree structures. And we went through existing models. This is a very powerful paper on optimal development of crypts, which had very deep mathematical analysis, and we were able to capture it very easily in ESTGs. And of course, the prey-predator model is easily captured, and you also get the lineage trees with bursts of the population that you can see here. And what else we did? Feedback regulation. I'll skip that example. It's fairly complicated. Implemented. I added a couple of slides, but I guess my computer crashed. Okay. So, okay, so I'll tell you in half a minute. That was the slides I got by email just this morning, but I guess the computer crashed and I don't have them. So we now close the loop, and let me see if I can just have a minute. Do I have a minute? Or a minute? A minute. Scientists, I really want to finish everything. Yeah. Tissue bank consent form, that's what my life is about. Okay. Okay, so we close the loop by using this tool. Finally, we have a tool that we can generate We can generate lineage trees on the computer with various interesting properties, sample randomly cells from them with mutations that are being constructed computationally according to known parameters, try to reconstruct the tree from these mutations and compare the tree that was reconstructed to the tree that was created by the simulation. And I remember why I wanted to show it, because this was an ESTG program that I wrote. It's the first program I wrote for the last 20 years, so I'm really proud of it. It's doing this simulation. And you get the trees with nice behavior, and you can use them. And, okay. So now I have to go to my other talk, because I have to say something very important. Happy birthday, Luca. Thank you very much.